morning or afternoon, everyone, depending where you're coming from. For those of you who don't know me, my name is McGinnis. I'm a hematopathologist at Vancouver General Hospital, and with Adam, I've been helping organize these OGM community practice meetings. So we have, I think we had close to 80 participants registered today. I'm close to 50 now, so it's a pretty good turnout. We have a couple items that we had scheduled on the agenda today. I'm just going to see if I can find the first person in here who I'm aware wants to present. But that agenda today includes a brief overview of some of the results of that survey I've been sending you over and over again for the last week on where Canadian labs are with OGM. A bit of a presentation from one of the scientists in our lab on our experience with the Early Access Stratus program. And then a bunch of really interesting case presentations. So a lot of exciting things to talk about today. We also have someone talking to us about their experience with EQA, and we're hoping we can lead that into a bit of discussion on how we should approach EQA in Canada. And I know that a lot of groups are interested in that, and that's something we should definitely be talking about more. But to start, just seeing in the participants pane, which is getting quite large, I don't see that Zaid has joined yet. So we'll give him a couple minutes to get in. It's early in the morning in Vancouver. And maybe what I'll start with is sharing my screen to show you the results of that survey I've been sending to the group with regards to what our labs in Canada are currently doing with OGM. And I see we have a good number of international registrants today. So we do have some international responses, and I can show you where that breaks down to on my share screen button here. Great. So is that showing up okay? Looks like it. So the survey I sent out, if you haven't seen it, please feel free to fill it out at any point afterwards or now. But it really just asks about where your lab is, what your lab's currently doing with OGM, if you have any instruments on site, what you're using it for, and what you plan to use it for. So you can see here right now we have.
It's a bit smaller. Can you make it any bigger? Maybe Control Plus.
Let's see. How's this looking?
Yeah, that's better. Awesome.
Better. Good. Great. Sorry, big screen in here. So first question for everybody is where's your lab? We have representation from all over the place. A lot of registrants from Montreal, Ontario, some from BC, and then a good number of international labs, as you can see here. And this all came in overnight, so I haven't had a chance to make it into a nice table format yet, but I can send it around to the group after the meeting. With regards to implementation of OGM, we have a good number here who are looking to learn about OGM, but really, a large proportion of people who responded do have either an instrument on the way or an instrument on site, and things are being validated or evaluated or reported. So you can see here, 30% or so of people who've responded, which is 8, are reporting at least one assay. And a lot of these are international respondents, with a couple Canadian respondents as well. A lot of people are in the process of validation. Sorry, just 1 second. I'm on service today, so please forgive me for stepping away perhaps. Okay, sorry about that, everybody. And you can see, again, what OGM instruments your lab currently using. Most of these are Sapphire. Obviously, this is the more readily available. No one's got just a Stratus, but 5 respondents have Sapphire and Stratus, and you should note this is some duplication. So I know a couple of these are our lab, one's BC Cancer, and then we do see some from other sites. Most labs that are validated and live are offering this for myeloid indications and other hematology indications. These 2 are VGH, and then others that are offering it or plan to offer it. Again, largely hematology, because that's a fairly easy space to access specimen for, but as you're seeing, constitutional disease begins to enter the field here. When looking more to the future though, you see again, overrepresentation in hematology, but solid tumors, prenatal, constitutional, adult infertility, repeat expansion testing, HPSCs. So there's a lot of interest in developing things across the board in OGM. A lot of this is, again, Canadian labs, but there's some international labs here. And I can show you the short breakdown of what those Sorry, just making sure that Zaid's able to get in here. Okay, good. Sorry for the disruptions, everybody. So moving back here, most of these labs that are clinically reporting testing are international labs. And you can see some of these are constitutional, some of these are CLL, constitutional, and FSHD in the case of an Ontario lab, and acute leukemias currently for our lab. So really, I think, takeaway, a lot of labs are bringing OGM on, and a lot of labs are moving along the validation pretty quickly from conversations I've had with some people in the country. And really exciting time to be in OGM and seeing where people are taking this, but a good time to start establishing common approaches and collaborating where we can. So if you haven't responded to the survey yet, please do. I'll compile these results once I've stopped receiving them and send a more meaningful summary of what we're seeing across Canada and to those international respondents internationally when I've received those. But these are still rolling in, as you can see, we're now at 29. So any questions about that short survey to date?
Hey,
Trying
Eric.
to get Zaid
Yeah.
in. Oh, there he is. Okay, look at that. I can't find you. There you are.
For some reason, I used I accidentally had Tara's name, I think, because I used her link to get in. Sorry about the confusion there.
That's okay. Thanks for coming in. So we just chatted a little bit about the survey that we sent out. So are you able to share your screen, Zaid, to chat about the Stratus data, or do you need a minute?
Yeah, of course. Happy to.
Great.
So while I get set up here, I'll just briefly explain a little bit of the context. We currently do have a Sapphire instrument in-house, validated. We've activated OGM as a clinical test for newly diagnosed acute leukemias. But we recently acquired a Stratus. And so I will touch on things about the Sapphire, but I think overall, I'm just going to try and give a short, maybe 10, 15-minute presentation summary about our inter-platform comparison. We really just want to highlight the differences that we've found, specifically with our heme testing, the differences between the Sapphire and the Stratus. And in order to do that, we had to do a clinical verification of the Stratus with samples that were run on both instruments, and we kind of looked at it from different lenses. So I'll just get started. As you're already aware, OGM is a technique that captures a wide range of variants in the structural variant landscape, with a resolution of up to 1,000 times higher than some standard of care methods. But there are exceptions, of course: haploidy, polyploidy, whole arm rearrangements with centromeric breakpoints, and of course, single nucleotide variants cannot be detected by optical mapping. How it works, briefly, I'll mention, because I'm sure you're all familiar with that. Our setup specifically involves taking bone marrow or peripheral blood in EDTA, and we freeze it and immediately after collection at -80 degrees, and then we perform the 1-day DNA extraction, the 1-day DNA labeling protocol, and then about approximately 1-day electrophoresis and imaging before generating some working data that we can begin to start looking at translocations, inversions, copy number alterations, the things that we know OGM can detect. So the Sapphire arrived in our lab in 2022, and we completed the end-to-end validation for it in early 2023. And for this validation study, I'll spare many of the details for. I did actually present this talk at ACMG, so that's partly why there's a reference for that here. So just ignore that for now. Basically, we did an analysis of 83 patients from a wide variety of hematological indications, including AML, MDS, BALL, one mixed phenotype acute leukemia, and 22 CLLs. We looked at standard of care tests received, namely FISH, karyotype, or FISH and karyotype. And we assessed the concordance for 129 variants assessed by standard of care testing. And then we also froze down samples within 4 days of collection, as I previously mentioned. So in summary, sensitivity, specificity, reproducibility were all above 97%. We completed this in mid 2023, and we started actually using OGM as a frontline diagnostic test in November 2023. So just approaching 5 months now. Now, given our experience and success to date with the Saphyr, we were pretty lucky to get the Stratus instrument quite early. I think we were one of the first 2 or 3 sites to get it, but I think we were one of the first sites in the world to verify it. And so this Saphyr instrument, shown here on the left, that's the one that we have in-house. This is the Stratus on the right. The first difference that I'll just highlight for you is the fact that the Saphyr has a flow cell capacity. Well, it accepts chips that have 3 flow cells, right? You can load 2 of them, so 3 to 6 is kind of the capacity there. The Stratus takes individual chips that have single flow cells, and it takes 12 of them, plus it has room for 3 additional ones that can be considered stat in a sense that they jump the queue or that they can be prioritized in the middle of ongoing runs, basically. So I first just want to highlight some similarities. Obviously, most of the wet work is nearly identical, with the exception of DNA labeling being about 25 minutes longer. But there's a slightly different chemistry, and so they recommend kind of not interchanging. We haven't actually validated or confirmed if that's true, but it seems to be the case that there's a slightly different protocol in that there's actually just the absence of one reagent during the labeling step, and they say that this is Stratus-specific chemistry and this is Saphyr-specific chemistry, despite the fact that the kits, I think, are nearly identical. It's just a difference in a few reagents. Now, the key differences here mostly lie with loading, imaging, and analysis. As I just mentioned, there are 12 flow cell spots plus 3 stat spots that can be prioritized over ongoing samples. Imaging seems to be slightly faster. It's a little hard to put an exact time because we haven't run that many samples yet, and we're actually still sorting through some of the timestamps. But for analysis, instead of using the Saphyr's RVA, which is analogous to reference-based assembly, for those of you familiar, the Stratus actually uses something called guided assembly, which is different in that it's almost a hybrid approach between reference-based assembly and de novo. So this takes a little bit longer than the RVA. It takes about 12 hours, which is the bottleneck, of course, when you have 15 samples running and you only have 1 server. That naturally creates a huge bottleneck relative to the Saphyr, with obviously the capacity to improve throughput if you're able to kind of scale the necessary parts. So keeping in mind that you would need to add multiple servers in order to significantly improve the throughput of the Stratus compared to the Saphyr. But right now we're currently bottlenecked with about 1 server, and so with a longer analysis time and only 1 server on our Stratus compared to 2 that we have for our Saphyr, it's a little bit slower. But again, the guided assembly, as much as it's longer, it also is it's a different analysis approach, right? It's being a hybrid between de novo and reference-based assembly. I think we've noticed some very interesting key differences that are going to get highlighted a little bit later. Our verification study that we performed on the Stratus, we took 22 patients that were previously run on the Saphyr, specifically patients chosen to reflect a wide variety of success rates previously seen on the Saphyr. So we chose about 86% first pass success rate of the 22 specimens. And this included 13 AMLs, 3 MDSs, 1 BALL, all bone marrow samples, as well as 5 CLLs, peripheral bloods. Also frozen within 4 days of collecting EDTA. And we analyzed the data for these 22 and found, and of course looked at concordance between standard of care tests received, as well as concordance with the Saphyr. And overall, there were about 31 variants with the following breakdown by class that we were able to look at. So our first thought for comparing these instruments before really giving some high-level statistics or interpretations, I think it's good to look at the raw data. So this is just the raw QC data for the 22 patients that were actually run on the Sapphire. So basically what you can see here is the identifier in the first column. Second column shows 19 out of 22 working on the first attempt, or a first pass success rate can be calculated from this. The next column, sorry, I should add the three that were the three that failed at a first pass were eventually passed after multiple repeat extractions or repeat labelings or repeat simply just running DNA on a new flow cell. The third column shows that there were 2 cases with discordant or discrepant results that were partly resolved. And again, we'll get into why those were discordant, specifically a TP53 loss and another TP53 loss and what we thought was a trisomy 12. And then this next column is again showing our end success rate was 100%. All cases were able to get good data, and then here's the raw data for your eyes to see. We mostly use N50 label density, effective coverage, map rate, and then positive and negative label variance to dictate our optimal runs. But again, 3 of these needed a little bit of extra effort to get that optimal data. So this is the data for the Stratus. Again, a few things to keep in mind before we get onto all the yellow and red. So we just received this instrument. And so naturally about 6 specimens did not work on the first try. We then re-extracted, again, following the same kind of troubleshooting approach, up to a maximum of 3 attempts was always our limit, both with the Sapphire and the Stratus. And with those 3 attempts, we were able to either generate optimal or suboptimal data for all the patients, except for one. One just failed repeatedly, and we couldn't really resolve why. And so our first pass success rate can be calculated from here. Our kind of end point, final point success rate can be calculated from here. This column shows similarly that the 2 cases with discordant or discrepant data were identical. But again, there were some subtle differences in analytical sensitivity that are key to highlighting the main differences between these. Now, why these are red and yellow are just visible here, right? We had one slightly low N50, again, really just variability in sample handling, DNA handling. We weren't able to collect enough data here, and that was still good enough to generate concordant data. So, they were still suboptimal. And a third one as well. And this was the only one that seemed to just fully fail across the board that we couldn't get any good data on. What suboptimal means, I believe I define on this slide, which I'll just summarize briefly. We go 10% of our QC threshold. So basically 210 kb is our optimal threshold for N50. We go 10% below that, 189. If there are 1 QC metrics exceeding 10% tolerance, then we consider that suboptimal. Or if there are multiple QC metrics exceeding our optimal threshold, but still within 10% tolerance, then it's also considered suboptimal. So I'll just hop back here for a second. Here's a head-to-head comparison between the QC metrics showing all the QC that were mostly in alignment between instruments. Again, the N50 being marginally lower can really just be attributed to sample handling and whatnot. The map rate and label density were also in alignment. One thing to note is that the 2 cases here those were part of the failed run that seemed to just generate no data. So of course there's a map rate that's low and a label density that's low, but everything else was more or less in agreement and in the optimal range. Effective coverage was good, with the notable exception of the cases that were suboptimal. And then PLV and NLV were again somewhat in alignment, with the Stratus being marginally lower for positive label variance and marginally higher for negative label variance, all still within our optimal range, right below 15%. So as I just said, these were the 6 QC metrics that we used. And above that, where am I? We defined optimal, we defined suboptimal, and we defined our failed cases. So that allows us to basically take our first pass success rate as being the percentage of patients to generate optimal data after up to 3 attempts, which technically for the Sapphire was 100%. Those are all 22 patients were able to get good data, whereas only 17 out of 22 were able to get optimal data. That's assuming we're talking about a success rate for optimal data. We, first of all, expect these things to improve with time. There's, again, we have a new instrument here. We've only been running it for a few weeks. There are some possible explanations that I'd like to discuss. The first being mechanical. I think that having a new instrument with this much flow cell capacity means there's a lot of robotics inside. There's basically a rotating carousel or a lazy Susan that rotates and allows you to kind of add flow cells to it. And then there's a little robot that's constantly pulling flow cells off. And so we've had some minor mechanical issues. So it is kind of an evolving territory as a new instrument. And then I should add that the core flow cell is brand new, but how it's loaded is entirely different. And that's kind of created some issues. I think that the new flow cells basically are a bit more difficult because you have fluid flowing in multiple directions within this microfluidic device compared to the old Sapphire flow cells that are a little more unidirectional and a little harder to clog. So I just think that in itself can be a factor here. But again, just to not really focus on this for a sec, we can actually dive a little bit further into success rate and break it down further by first pass success rate. Did we get good data on the first attempt? And endpoint success rate? Were we able to get data after 3 attempts? But then even within those, we can break it down further to optimal and suboptimal, optimal and suboptimal success rates. And so the only 2 things that I want to highlight here is that the first pass suboptimal success rate for the Stratus was marginally higher with 20 out of 22 at 91% compared to 86. But really at the end of the day, the question is, we can always improve our expertise and our mechanical factors. Are we able to get good data? And this was about 21 versus 22. So again, still within alignment with each other, and we expect these things to become comparable over time. So the key purpose of this entire verification was to ensure that there's no loss in analytical sensitivity or specificity. And in short, I'll just spoiler alert, all variants detected by the Sapphire were also detected by the Stratus. But surprisingly, we identified a slightly improved performance in analytical sensitivity with the Stratus, which I'll get into. First, I want to highlight the cases of standard of care variants that were missed by OGM. So first, we had a case that was optimal by the Sapphire, suboptimal in the Stratus due to a slightly low N50 that failed to detect a TP53 deletion in a CLL in 22 out of 200 cells, shown here on the right with this fish pattern of 1 red, 2 green. And here on the left is the copy number track for 17. And some things to keep in mind, 22 out of 200 cells, that's 11% of cells. Our cutoff for a lower limit of detection for our cytocell TP53 probe is about 8%. This also has an expected VAF by OGM of 5.5%. So we're right at the limit of detection for what we would expect for OGM. Which, the fish that was performed here was also cultured on cultured cells for 24 hours. So it's also hard to say exactly what the VAF would be, but of course, keeping in mind that we kind of need to consider this a false negative call for our calculations of sensitivity and specificity for both the Sapphire and the Stratus, a true missed call. So the second case that I wanted to get into was interesting because it was detected in 30 out of 100 cells, or 30%, with an expected VAF of 15%, well above our limit of detection. But interestingly, this was not detected on the Sapphire. You can see right here that there are 2 copy number calls actually directly flanking the TP53 locus. So technically, this TP53 is not deleted. Obviously, we've recognized that this is actually a very common pattern in this region and that we would typically consider this a provisional detection. But again, just for the sake of keeping our analytical sensitivity and specificity calculations standardized, we included this as a false negative. for the Sapphire. Using the guided assembly approach, though, you can see that we saw a unambiguous, clear copy number loss of all 17p. This was a true positive call for the Stratus, and this difference we really kind of attributed likely to the guided assembly approach. And then there was a third case of a trisomy 12 in 52% of cells that we actually didn't see anything unusual for on either instrument. But we investigated this one a little bit further. And although no metaphases were available for karyotype, we were able to do FISH on another marker on chromosome 12 on ETB6 that did show 2 normal signals. So no cells demonstrated 3 signals. We concluded that this was probably that this finding upon performing FISH initially using a 12 centromeric probe yielded a false positive finding, probably due to some focal gain of the 12 centromere region, which we know OGM would not pick up, and that the trisomy 12 is actually not really a true call. So we omitted this from our calculations. And then fortunately, this false positive FISH finding would not impact patient management as well, given the concurrent TP53 deletion in patient 17, as well as a proximal loss of 13q region that was also detected in this case, both by Sapphire and by Stratus. So overall, the sensitivity and specificity were comparable here. We have about 100% in the Sapphire and the Stratus for specificity, and then 94% sensitivity for the Sapphire, 97% for the Stratus, owing to that one TP53 deletion being the difference there, right? So again, everything is still in alignment with just marginal improvements that we found with the Stratus, despite the fact that maybe our data may not have been as optimal as it was for the Sapphire. Now, I also just want to break this down a bit further by instrument as well as by class. So you can see those 2 core values here. The loss in sensitivity comes from the deletion calls. As I mentioned, the one TP53 deletion that was not detected by either in 5.5% VAF, and the other TP53 deletion that was detected by the Stratus but not by the Sapphire. So a couple other differences I just want to highlight. We were able to compare the VAF assigned by the Sapphire and the Stratus for concordant SVs and copy number calls, and showed that on average there was about a 2% difference in that the same variant across instruments was 2% lower on the Stratus, with clearly the bulk of more significant differences being with translocations shown here, right, up to 20% different, or 20% lower on the Stratus. I also wanted to highlight another small difference we found. Basically, what you see here are the total variant calls in a given case for high confidence and for low confidence across the Sapphire and the Stratus. Of course, the high confidence calls were nearly identical at about 60, 70 variants each, whereas the total variants were actually reduced on the Stratus, which we suspect likely due to some kind of guided assembly approach, perhaps an improved control database. Little unclear, but effectively what this means, or the key takeaway, is that there are fewer low confidence calls with the Stratus with guided assembly. So all in all, to summarize, we seem to be one of the first sites to have this, and we have completed our verification and will likely begin using it soon. Our findings, like most, of course, demonstrate the utility of this instrument in providing a unbiased, high-resolution whole genome evaluation. We've already started using the Sapphire for clinical for newly diagnosed acute leukemias and are looking to expand indications also in the near future. And we will be running karyotype in parallel, but we'll be phasing this out over time. So with that, I'm happy to take any questions or yes, Susan, I see you have raised your hand immediately.
Hi, Zane. Good to see you again. Thanks. I love this talk. It's really well done. Thanks again for that. I just wanted to ask quickly, for your patient number 17, when you did the comparison between the Sapphire and the Stratus, For the loss of the 17, did you run that through VIA with their improved calling algorithms? Because we found that we now see things that were invisible just on access with the Sapphire, that through VIA, those calls are being made at a greater level of sensitivity.
We don't have VIA set up.
Okay.
Are you saying VIA has like a different, is it it's analyzing your molecules in a different way, different than guided assembly?
Yeah, the statistical algorithms are greatly improved for SV and for copy number calling. So we found that those invisible losses of TP53 that they weren't detected before, that they are visible now with the VIA software.
Oh, okay. Well, yeah, I didn't know that. That sounds amazing, and yeah, we'll definitely look into it. I'll keep you posted what happens, but
Perfect.
I see there's a question just about the trisomy 12, which I suspected, and I also failed to mention. So I'll just go back briefly to mention that yes, the third signal here is smaller, and we did note that and suspect something was a little funny, but only upon kind of retrospective analysis and downstream testing that did we recognize that yes, it probably was a artifact of some focal small centromeric amplification of this probe and not a real trisomy 12. So to clarify, this was not included in our statistics because we considered it to be not a real fish call for a trisomy 12.
Thanks, Zaid. That was a great talk. I haven't had the chance to see it yet, so I'm really happy I got to see that today.
Perfect.
For those who haven't met Zaid, he's done a lot of great work on OGM with our lab, and he's presented nationally, internationally with this. So he's doing a lot of great work advancing this for us.
Yes, thank you, Eric. You actually just reminded me, I probably should have, I think I forgot to get to this. Of course, my acknowledgement slide before I hand it over, I need to thank the team because I do work with a great team at Vancouver General. I, as a lab scientist, get the pleasure of working with geneticists like Tara, pathologists like Eric, and lots of great people that are also geneticists or medical technologists. So I just wanted to thank them all, and of course, thanks for giving me the chance to speak, Eric. I'm always happy to share interesting things.
Great, thank you. Yeah, Susan, thanks for bringing up VIA. We've been trying to get it installed, but with our security scans ongoing for our new instrument coming in, finding time to get them to actually get in there and do it has been challenging. I wanted to ask, while we're on the topic of Stratus, I know a couple other labs do have Stratus on site. Is anyone interested in sharing their experience and how it's performed for them? And if not, that's totally okay. Okay, great. Yeah, we're excited to have it on site because as you heard from Zaid, we're using this for acute leukemias right now. And if we end up getting more specimens than it can handle, we're stuck with waiting for those 3 specimens to finish running, or do we pull it off and start a new one? So having the opportunity to bump things on a stat cell, really appealing. And anecdotally, haven't yet timed it, but watching those specimens collect, they seem to go a lot faster, at least in the data collection phase. And so I don't know if that's just eyeballs saying that looks faster, but when we loaded 15 specimens on there, it seemed to be like they went through a little faster than I would have thought they would. I see Adam has his hand up.
Yeah, Eric, I wondered about, and Zaid too, maybe, are you guys what are you using for your computes? Are you using the onboard on the Stratus, or are you still using the servers, the compute clusters that were shipped with the Sapphire?
So the Sapphire compute is still dedicated to our Sapphire. So we're using the onboard Stratus right now for Stratus.
And how are you finding the compute time?
Like Zaid said, it's a little bit of a bottleneck if we have to use it as a stat indication. So, we're toying with different ways to approach it. But we do hear it will decrease over time, which we're looking forward to. But in the current iteration, the Sapphire does do a pretty good job of pushing things through quickly. The Stratus, we're not sure how it'll do in terms of if we get 4 acutes, we have to run them all and see how it goes. So it's a little longer, for sure. I haven't been able to pull data off of it to say exactly how much longer. They estimate 12 hours or so for us, which seems accurate from what we've seen. But again, don't have times on it. We have
Yeah.
talked about whether we need to get another server to help support that compute, but that remains to be seen.
Yeah, I should add, technically, we really only have data right now on the length of one run, because I think when I started the first sample, I had the starting time point, and then I had the ending time point, and it was about 12 hours as expected. But I think everything else kind of got, I think when you're running more samples, all of them take longer, and then that kind of throws off the timestamps a bit. And obviously it's a moving target. It takes longer to run 10. It takes longer to run an individual one when there are 9 others than when there's just one. With a bit of and we haven't really mapped that out yet. But I would be curious if anybody has any insight on that, what they think.
Well, and I also think, I mean, I know that the onboard compute time for the Sapphires is just going to get better and better. I mean, I think the idea was that you was going to get down to about 2 hours, which would be, ideal. Like, you'd be able to have that sample come off, because if you've got slots for 12, right, you've got 24 hours in a day. If you can basically crash through, 2 hours per sample doing your assembly, then a whole day's run you can be able to run through in a day. So I mean, let's I know it's not there yet, but hopefully it will get there soon.
For sure.
The other consideration for groups who have both the Stratus and a Sapphire is that the Stratus, as they touched upon, uses this guided assembly pipeline as the exclusive approach, and Sapphire historically hasn't had that approach. So we're in the process of updating our Sapphire, but like I said, we're dealing with some security scans, and we need to arrange downtime for the clinical testing at this point for that update. So we haven't yet had a chance to try guided assembly on our Sapphire server. But if you have an older server, we've been told it works, but they don't know how fast it works. So we have yet to see, if we want to increment to guided assembly on our Sapphire, what's that turnaround time going to look like? So something to keep in mind if you've bought a server and it's older. Great. Anything else on Stratus, or should we move on to some? We have a nice full schedule of cases and some experiences with EQA internationally. So if there's no other thoughts on Stratus, we can go ahead with that. Oh, sorry, I see a question in the chat here. Isabel, have you tried running a guided assembly with Sapphire data? So what we were told, so we have, like I said, we're trying to update to the new access version so we can do that guided assembly. That's the question, is it machine related, assembly pipeline related, both? Hard to say. I see Melanie has her hand up. We haven't yet had a chance, no.
Yes, we have started running guided assembly on the Sapphire. I can't say much because we're in the early stages, but we also did see additional abnormalities that were not detected by the RVA. But again, it's very, very limited data at this point, so I can't really speak more to that. One thing we did notice is that we had to upgrade the server in order to install the latest version to accommodate guided assembly. And when we looked back at the RVA on those samples on which we also wanted to run guided assembly, we noticed that the update seemed to have changed something in RVA. And so we were now seeing a few things by RVA on the same sample that we had not seen with RVA using the previous server. So I don't know if something changed in the background, and this may be just anecdotal. Again, we've only been doing a comparison for, I think, 3 or 4 cases right now, but it seems like the upgrade may have changed something. So I do wonder, when Susan mentioned that things were called with VIA that were not called before in Access, I wonder if it's something specific to VIA, or it's because VIA runs on an updated version of the server, and this changes things. And I don't know exactly what's the effect of all of this. And I haven't had a chance to talk to anyone in BioNano to investigate this further. But anecdotally, we did notice some differences for the same pipeline, but using different versions of the server.
Well, thank you for sharing that. That's important to know because we're considering, like I said, updating our server, and we have a bunch of clinical cases sitting on there. So it would be helpful to know if things are going to change in the specimens that are already done.
Yeah, and I think it speaks to the point that whenever you update anything on any of your instrument or pipeline, you need to do a validation, because surprise, surprise, sometimes either the company doesn't realize that it impacts your testing, or they don't tell you, but it can have an impact. And anecdotally, it is indeed like deletions that we saw that had not been picked up before. So it may speak to Susan's point that they were seeing TP53 deletions that they were not seeing before. I don't know.
Susan's got her hand up again.
Hi. Yeah, so we've done some comparisons so we can share at a future time with pulling all the calls from the back end for 1.7 to 1.82 via and soon to 1.8.1 too. So we'll have some of that comparison, and that's for in excess of 50 cases with the comparison. So we can do some discussions around that at a future time point too.
Great, look forward to it. All right, it's a great discussion. Maybe to make sure we get through the things we've scheduled for today, we can go ahead with one of our next speakers. So we can perhaps go ahead with the cases for the time being. I think it's about 10 minutes per, so we should have a little bit of time to get through those and then have a discussion about EQA with Victoria Marcu from Israel. So I have 4 people who I understand confirmed presenting cases today. And I'm going to pick in no specific order from the list I have here. Is Fenny from Sherbrooke available?
Hi, yeah.
Hi. Perfect. So I think you should be able to share the screen. So please go ahead. When I see the title of your talk is Interesting OGM Cases from Sherbrooke.
Good. Can you see it in full screen?
Yes.
Good. So hi, everyone. I'm Fanny Thuriot, and I'm a clinical specialist at Sherbrooke. Just to let you know, we've started a little bit of the validation, but not yet completed, mostly for MDS and AML cases. So here I'm going to show you 2 interesting OGM cases that we had so far. So the first case is an AML patient with 50% blasts, and I hope you can see here the karyotype. So we did a karyotype, and we had this little derivative, if I may call, chromosome 21. So at first we thought it was a deletion of the terminal short arm of 21, long arm of 21q. And as you all know, there's RONX1 that is associated with AML in this region. So we decided to perform FISH with the RONX1T1 and RONX1 probe. And I hope you can see it well, but we have 2 signals for the RONX1T1 from chromosome 8, so that's good. But we have 3 signals from the RONX1 probe, and 2 are smaller, so there's a DIM in 2 of the signals. So this is not a deletion after all. So we decided to report it as a derivative chromosome 21 resulting from a translocation of chromosome 21 and a known partner. And we also performed CMA, and this was a normal result. So we did run OGM testing for this case, and we were able to confirm the partner for the translocation. So it was a translocation between chromosomes 14 and 21. So we just switched the chromosomes here. So the derivative 21 that we thought was actually a derivative 14, and the chromosome 14 that we thought was actually the derivative chromosome 21. And it's a little bit small here, but we can see that there's a rearrangement between SAL2, which is on chromosome 14, and RONX1 on chromosome 21. So it disrupts that there's really a break in RONX1. In the literature, the fusion partner of SAL2 and RONX1 is not known yet, So there's no known biological effect, but maybe in the future we'll be able to characterize more this rearrangement. So with this case, we were able really to confirm the rearrangement and confirm the partner of RUNX1. The number 2 case is also AML patient, and we saw an inversion 11, so pericentric inversion with breakpoints estimated to P15223. And because it was close the region of KMT2A or MLL, we did a FISH. Results are not shown here, but it was normal. So we decided to run OGM on this patient in order to see which partner could result in a fusion. And we had OGM, so we confirmed the inversion, pericentric inversion of chromosome 11. The breakpoints were really close than the karyotype we did. And we had a rearrangement between NUP98 and DDX10 that we can see here with the diagram here. It's a known fusion, but it's really rare, and unfortunately there is no prognostic value for this one, but we classified it as intermediate prognosis for this. So that's it. Those are 2 cases that I wanted to show you, and if you have any questions, I'll be happy to answer.
Really interesting cases. Thanks for sharing those. Yeah, I think the things we're seeing are very surprising in terms of, yeah, I wasn't expecting that, right? Have you seen, so the RUNX1 breaking in the kind of intergenic region of the cell2?
Yeah.
We've seen that, I think, more with MECOM, but I honestly can't think of having seen it a lot. Have you encountered that a lot with your OGM data? Because I know one of the things we're looking at is, you know, how frequent that is, which we haven't seen a lot of it, but sometimes the gene bed files that are provided with the Sapphire access differ from other coordinates in the same genome build. So particularly the ones that include more of their like known canonical transcripts, sometimes like KMT2A shifted or ABL1 is shifted. So you've seen base or ABLs where it doesn't break into ABL1 on the BioNano provided gene list, but every other gene list you download does. So have you looked at that?
No, I'm pretty new with the OGM. I just started working in Sherbrooke in January. I just finished my training, so I'm just looking around the OGM cases. But it's pretty interesting. I will look at that, surely.
Great. Yeah. Any other questions about those? I think those are really good examples of why we need this, because you never, I mean, unless you know exactly what you're looking for, you're never going to find it. In the RUNX1, you did find it. But the NUP98, unless you have that probe and you're going looking for it.
Yeah.
Yeah. Great. Well, thank you for sharing those. Those were really interesting cases.
Thank you.
So there are no other questions for that one. Perhaps the next one I have in the list here is Sakthi Morthy, identifying chromosome 20q11.21. Chromosome 11 is. Hello.
I can
Great.
share my screen. Are you able to see my screen?
Yes, thank you.
Awesome. Hello, everyone. I am Sakthi Murthy. I'm a lead scientist at CCRM. CCRM is a not-a-profit organization. It's a CDMO. It's located in Toronto. So we are not in clinical laboratory diagnostic anything. So I don't have any data looking into the OGM, but we are exploring to understand some of the chromosomal abnormality that we often see when we culture induced pluripotent stem cells. So we are further downstream in terms of therapeutic values, like understanding how iPSCs can be leveraged in bringing out cellular-based therapeutics for some of these diseases. So I will be quickly talking about
sorry. Can I just interrupt you for 1 second? It's Adam. Yeah, I just wanted to just say to my cytogenetic colleagues that this is sort of what's referred to in the OGM community as cell bioprocessing, and it's an area that I think a lot of places we've been traditionally using karyotyping to to look at these cells that are going to be used for therapeutic purposes, and to look at the culture effects, etc. So it's something that probably a lot of labs don't have experience with, but it's a very interesting area, and I think that the potential for OGM is really interesting, which is why I thought it was really nice to have Sakti present at the meeting today. So I just wanted to interject that so everybody kind of knows where we're coming from here.
Thanks, Aaron. Thanks for the introduction. Really appreciate it. So what we are doing in terms of iPSCs are iPSCs are pluripotent stem cells that has the ability to differentiate into functional any cell type. So there are two types, like autologous, like where you use the patient cells and then you reprogram it, bring it to iPSC, and that can be further differentiated into different cell types. For example, the good example would be like a CAR T cells that can be used for cancer treatment. And the other approach is using healthy donor cells where we can reprogram into iPSCs, and then further it can be edited in a way that it can be immunoclocked so that you can develop like universal donor cell line that can be used for any patients. So it will be like off the shelf, like where it has all the required elements to have the cellular therapeutics, and then it can be administered to patients. So when we go by the allogenic cell therapy route, then it's very cost effective because it's off the shelf, as one universal donor cell line is be enough to cover the entire population, and we will be screening for biologically fit cells. That's where we are looking into understanding the chromosomal abnormalities, everything in each process before, because they goes for the therapeutics. And we also looking for like there will be no donor variability because we all be being tested, all the differentiation process can be optimized, everything on one cell line. So there's no need to optimize for each and every patient at a time. So the major challenges in terms of safety perspective at genetic levels is a chromosome abnormality that we see in these iPSCs, because these has to be cultured, grown in long-term in culture, and these cells can acquire these ability as a strategy to adapt for the cell culturing processes. And we also see some of these ones during the processes, for example, reprogramming, where we have to do a whole epigenetic modification for these cell lines to bring from a fibroblast or CD34 positive cells to induce pluripotent stem cells. And also during the process of gene editing, where it can be induced by the CRISPR-based system, where there can be some chromosomal abnormalities or arrangements can happen because of these processes as such. So these are the list of commonly occurring chromosomal alterations that have been studied so far, and it's been seen in human pluripotent stem cells quite often. And the one major one is chromosome 20, Q11.21, is shown to be occurring in at least like 37% in the known existing iPS cell lines. It's so robust, and it has also like a growth advantage that these kind of quickly can populate in the in the growing cells, and then it can just overpower the growing cells, and it will be the most dominating genotype in the cell culture. So what that would affect? So obviously it affects the differentiation potential of pluripotent stem cells. So it kind of will not differentiate to all the cell types. Certain lineages is not possible when you have these particular type of gain in chromosome 20. Also, these also kind of activate some of the oncogenes, BCL2-L1, which is known to have some, and it helps in mitotic survival of these cells. So obviously, in a regulatory perspective, we don't want these kind of abnormalities that's persistent in the therapeutic cells. Obviously, we have to constantly look for, screen them, making sure that these type of abnormality doesn't exist in any of these therapeutic cells. So for the case study today, like I'll be presenting you some of this data that we have generated using different approaches to understand identifying this particular abnormality in iPS cell line. So one thing we noticed during our gene editing process that the parental cell line had a normal karyotype in G banding, but they seems to have only in the edited ones, few cells seems to have this abnormality. So we asked the question, is this abnormality due to a gene editing process, or cell culturing process, or does the parental cell line as such has this particular abnormality? So these parental cell lines, when we do an organ karyotype by G-banding, which is always like a gold standard for understanding or characterizing this pluripotent stem cells, we always look for 20 cell counts, and we always see them to be having a normal karyotype. And particularly chromosome 20 seems to be very submicroscopic, that we can't able to detect them because they are having like a smaller size in the gain. But when we did the fish analysis, what we saw was about like 91% of this population had a normal karyotype, like a diploid. Only 7.2% of this population had a gain at the BCL2L1 region. So this shows there is like a gain in mosaicism, and it's also like even fish has a restriction because of the length it can detect, and also depending on which region is duplicated, you'll be able to detect only certain cells having that particular duplication. So as this particular duplication is very recurring in the iPSC world, so there is also kits that can detect them at a minimal duplication level. So using like a ddPCR based assay probes, we could be able to detect them at a minimal like a 400 base pair differences. So when we used this one, like we were able to see that even the parental cell line had a minimal duplication. So here the graph here shows the purple bar over here seems to be like all the regions gaining the 20Q11 2.1 at this region, whereas compared to the control DNA, which seems to be having a normal karyotype. So what we saw is this particular gain didn't come during the process, but it's already been in the parental cell line, which seems to be a very minimal, which kind of can't be detected using a karyotype G-banding, but could be detected by a very sensitive method like a ddPCR. So the one which I'm showing is just an example, but we have cases where we had instances like using a normal karyotype G-banding, there will be like non-clonal population, and we kind of process those cells, but eventually during the culturing process, they would have kind of dominated the cell lines, and then we'd have seen them later in the process. So this would give us like a obviously expensive culturing process we just go through, and at the time of kind of clearance, we will be seeing that, oh, we can't use that further down the road. Then we have to again like discard these cell lines. So it's always better to have more sensitive methods so that we can detect them at early stages of when we're able to see those abnormalities harboring in the cell lines. So further, we also investigated to know like whether these differences or the gain in these regions, does it have any functional importance? So it has been shown in literature this region is more required, like there are regions that it could activate oncogenes because of this particular duplications. So we were looking at the control and the iPS11 cell line, which had these particular duplications. So this is like a single cell RNA-seq analysis data. And we can see like overlapping of these single cell reads, we can see there is like differential expression patterns coming from these cell lines. However, there is like regions where we can see like increased expression of certain oncogene markers like TPX2, BCL2L1, and ID1, kind of having like certain populations seems to be having like 10% or something, having like higher expression. So eventually when these cells are going to be cultured for longer time in culture, we could see that these population of cells kind of dominating, and then there will be increased expression of these oncogene panel. So our future goal is to understand advanced technologies to understand these chromosomal alterations that can occur in routine culturing or during any processes. So we are interested to implement the OGM technology in our pipeline to understand whether this can be used as a routine cell characterization, or for each programs, like even for iPSC, we do reprogramming, editing, and even scaling up to master cell banks and differentiating them to different cell types. So we are kind of interested to understand this technology more to implement that in our pipeline. That's all from me, and we'll be happy to answer if you have any questions.
Great, thank you very much for that presentation. Very interesting and a really neat field that's exploding. Sorry, I missed part of it because I'm dealing with some clinical cases at the same time, but do you see this testing being, mostly valuable for designing processes, or are these done as kind of quality assurance before products are released to patients, or both?
So usually what we do is like for each process when these cells go through, for example, they start from reprogramming. So we always do a characterization of the cell lines to make sure that they are iPSCs, they are karyotypically normal. So most of the times this kind of missed because the gold standard is just using the G banding approach. And we often miss these chromosomal abnormalities in the early stages. And when we go to the gene editing, that's what we find, oh, there is a abnormality that would have been in the parental cell line. So the goal is to have them implement in routine cell culture, like every 5 passages or 10 passages, and eventually maybe implementing them for each and every processes when we do, like from reprogramming to gene editing and further scaling up. So having this advanced technology would help us to have a quality assurance of these cell lines down the road so that we are comfortable knowing that we're not looking or working on a cell line that is having some chromosomal abnormality.
And again, apologies if I missed it, but if you do find a chromosomal abnormality in cell culture, then what's your action? Do you toss that?
Mostly they can't be used for further processes because it would be flagged for the regulatory guidelines and it can't be used for therapeutic purposes. Even if it is kind of having any known functional significance or unknown, but any chromosome abnormalities, which is kind of not just once, then it will be flagged and then can't be used for therapeutic purpose.
Very cool. And then we see a lot of patients, or not a lot, but we're seeing more patients for CAR T-cell therapy in Vancouver, and that's expanding. Do you see a similar role in that kind of space, or is it less of an issue where there's, or do you see a similar role in that space?
I think for CAR T cells as its patient cells, mostly like maybe like once the process is done, like obviously there should be some safety analytics has to be done to make sure that like the editing process doesn't kind of create any new abnormalities like genetic alterations apart the one from the design. So there will be always like a safety checkpoint, but we have to see like how the regulatory works with the autologous compared to an allogenic one.
Right. Very interesting. Great. Thank you for sharing that. Any other questions for Sakti? Great. In the interest of time, I think we can move on to the next presentation. So is Aaron Marshall from CHIO available? Presentation titled Investigation of an Inherited Copy Number Gain in DMD by OGM.
Yeah.
Hello.
Hi. Okay. So hi, I'm Erin. I am a postdoc at CHIO with Care for Rare. And one of the things I have just started investigating is the use of OGM for constitutional cases. And this is just one little case that we've been looking at recently, which is, as you said, investigation of an inherited copy number gain in DMD. So this was a 3-year-old female that was referred to the CHIO Genetics Diagnostics Lab for developmental delay. It was seen that there were gains of exons 45 to 51 in the DMD gene that was detected by chromosomal microarray. So that was the call here. This was predicted to result in an in-frame duplication if the gain was the result of a tandem duplication within DMD. So this was at that time interpreted as an incidental finding of unknown significance. So the reasoning for this is because duplications of exons 45 to 51 have been reported in patients with Becker or Duchenne muscular dystrophy. So seen here is just the UCSC view, and in blue is highlighted the region that was called as a gain by microarray. And then here are some ClinVar copy number variants, and these are all, this is filtered for those that are path or likely path duplications and copy number gains in ClinVar. So this was investigated further. So FISH analysis actually showed that this DMD gain was paternally inherited and was actually inserted into cytoband 17q21. So that can be seen right here in the proband, and you can also see that here in the father. So the proband's father is also developmentally delayed, so this complicated the interpretation of this even more, because then it wasn't sure if this insertion was interrupting a gene. So yeah, given those unknowns in the exact insertion site, it wasn't clear if there were genes disrupted at these breakpoints. So we then utilized OGM to detect this DMD insertion. So you can see that here. So this is your chromosome 17, and you can see the insertion of DMD. This insertion is in direct orientation, so you can see that based on the genome location. This wasn't called as a CNB based on the thresholds we had, because it was just under the 500 kb threshold. And zooming in on this a little bit more, you can see that the DMD insertion is in the intergenic space. So these were the labels that mapped appropriately on chromosome 17. And then these labels weren't mapped here, but you can see that these are likely the same labels. And this is actually probably the location of the breakpoint, which is completely intergenic. And this exact same insertion was seen by OGM in the father as well. And in parallel, this was also sent to sick kids for genome sequencing. And you can see that our results actually correspond pretty well. So the chromosome X gain was seen by both technologies. Again, this wasn't directly called by OGM, but you can infer that based on the insertion labels and the copy number track. And the coordinates correspond pretty well. The insertion site again was mapped using genome sequencing, and you can see that these coordinates also correspond pretty well between genome sequencing and optical genome mapping. And obviously there's this range here just given on the distance of the labels. Genome sequencing was able to detect there was actually chromosome 17 deletion at the insertion site. So that entire 1.35 kb was deleted. And obviously that was not detected by OGM because that was within the insertion site. So there aren't really any labels that stratify either side of that. And both of these technologies show that there were no genes disrupted by this insertion. So the chromosomal, again, the chromosomal microarray only detected the gain. So it was very uncertain. And then the FISH was able to determine that it was unbalanced insertion. But again, we weren't able to get the resolution that we were able to by genome sequencing or optical genome mapping. So the conclusions here are that the clinical interpretation of these intragenic duplications obviously remain a challenge of chromosomal microarray. A follow-up testing with FISH can't resolve the exact breakpoints, orientation, or the exact insertion site. So that doesn't help with resolving clinical uncertainty. So OGM was employed to overcome these limitations, and it allowed the structural variant to be investigated at a much higher resolution. And importantly, this did also have good concordance with genome sequencing. So with that, I'd like to thank the patient and her family, the CHIO Genetics Diagnostics Lab, who did the clinical CMA of the FISH testing, and Dr. Jean McGowan Jordan and Dr. Melanie Bergeron are in charge of the OGM constitutional project that I've been working on, the care for our team and our collaborators. And again, it was the Hospital for Sick Children who provided the genome sequencing data for this project. And thank you.
Great, thank you. Very interesting case. Are there any questions for Aaron? At the risk of exposing how little I know about DMD as a hematopathologist, does this help you explain the patient's phenotype?
No.
Yeah.
So it was DMD is usually like a later presenting, so that's why I also wasn't sure if it was just some type of incidental finding. And it was unknown significance because it wasn't sure if it was a tandem duplication or not. Because it's not, and because it doesn't seem to be interrupting a gene, I think it's been downgraded even more. And we don't think it likely has any clinical significance, but time will tell.
So it's maybe the advantage of making things a little less certain or less uncertain.
Sorry, maybe I can interject here. This is Kathy Chen. We did the genome sequencing here
Yeah.
at SickKids. So we have a patient who had exactly the same duplication. Our patient was referred as a 1-day-old with multiple congenital anomalies. And we also found that the duplication was inherited from a phenotypically normal father in this case. And when we did the sequencing, we found that the breakpoints in both patients were identical, both on the X chromosome and on 17q, like literally to the base. We also ran it through a lot of in silico pipelines here, comparing identity and things like that to the common to the normal population, and found that it wasn't, the patients were not related. Because my first thought was, oh my gosh, these two patients are related and they don't even know it. But it turns out that they're not related. And we just recently learned at the ACMG that there's a third patient with what we think is exactly the same duplication. I've asked them to send their whole genome sequencing data to us so that we can also run that patient's information through our pipelines. But we think it is the exact same breakpoint as well. So I'm still looking for a common haplotype for chromosome 17 to see if these patients are, have a common haplotype, which we think they will, but just haven't found the SNPs yet. But I don't think this is pathogenic. It's just likely a ancestral rearrangement that happened a long time ago, and that's just floating around.
That's interesting that you found a third patient.
Great, very interesting. And
I think it's also really interesting that it's the exact same breakpoints. I wonder if there's any underlying sequence elements, like the 1122 with the palindromic sequence that mediates Emmanuel syndrome rearrangements. If there's something significant
Adam,
about those sequences that makes them prone to rearrange. Yeah,
we've looked.
Wow.
There's nothing. There's nothing. Yeah, and this third patient is from CHOP, so they're American. So it's probably, oh yeah, and this they're probably European. All three families appear to be, well, our two families anyway appear to be European. Yeah. The third one probably is as well. Yeah.
Go ahead, Melanie.
I was just wondering if, I suspect we're going to be finding more patients over time. And it does make me wonder, because, historically, when we saw those gains, either by microarray or DMD testing, not all labs would do the follow-up testing to determine if these are in duplications, tandem duplications or not. We know that most of the interstitial gains seen by microarray or MLPA are tandem duplications, but clearly some of them are not. So it does make me wonder, about any patients who may have been told that they are carriers of a DMD gene rearrangement, when in reality they may be carrying this rare ancestral rearrangement. I think not to brag too much about cytogenetics, but obviously good old cytogenetics replaced by newer technology like OGM, I think still has a place in the interpretation of these, and you cannot always assume that what you see is disease causing and fits with the phenotype, particularly if the phenotype is not specific and can fit with more than one disease.
You make a really good point, Melanie, because this third patient, well, first of all, to your point about duplications in DMD, if they're out of frame, I don't think we worry too much about it, but because this one was in frame, further investigation definitely needed to be done. Our third patient from the States, it's really funny. Mom had been told that she was a carrier of a DMD duplication. I can't remember the exact story and how she came to get it. I think she was doing PGD or something. I can't remember. Anyways, she was told that she was carrier of a DMD duplication. And then I think the patient was also female, if I'm remembering correctly. I can't remember. Anyways, so they were told that the baby was also a carrier of a DMD duplication until they went and did further testing, and it was sent to a company in Philadelphia, and they found, that it was actually an insertion into 17, and then they sequenced it and, found the breakpoints, et cetera, et cetera. So, yeah. Oh, I found my slides on the in silico stuff that we did. So we actually did IBD testing against our more than 10,000 host-seq samples that we have here at TCAG, and we confirmed that the parent-child relationship, but that the second proband was unrelated. So we knew that, and also what else did we do? Yeah. And then, sorry, I didn't know you were presenting this. I would have looked up our information a little more thoroughly. But anyways, yeah, we're still looking for the haplotype.
Great, very interesting case. And I'm sure we'll see a lot more things like this as we do OGM and find weird and wonderful things. But it's great to see, even at this early meeting, all this coming together and people sharing observations like that. It's fantastic. Thank you for sharing that. I have one more presentation for a case, which is Melanie, regarding OGM calling of recurrent gene fusion breakpoints in hematologic disorders.
Yes, let me share my screen. So hopefully you can see, not this. Oh, I need to switch. You'd think I know how to do this after 3 years, but apparently not. Okay. Yes. So I've decided to focus on one specific case of a pediatric BLL. So for those of you who may not be familiar with BLL, obviously lots of known rearrangements, and ETV6 RUNX1 in particular is rather frequent in pediatric BLL. About 20% of cases in children less than 10, and then 10% in children 10 to 15, and then it gets more rarer in adolescents and adults. So any lab who does cytogenomic testing for pediatric BALL is very well familiar with ETV6 RUNX1. The breakpoints are between exons 5 and 6 on the ETV6 gene, and this paper here, they labeled the exon as 1A and 1B, and so the breakpoint would be after the first 2 exons. Obviously, some transcript in the USC browser referred to as exon 1, 2, 3, 4, 5, and so on. But just bear in mind that the breakpoint is after the second exon. And the driver event is on the DIR21 of this 1221 translocation. So we have had a few cases of ETV6 RUNX1 that we were able to study by OGM. And this is what we normally get, where the breakpoint is somewhere in this large intron of RUNX1. So you have the first 2 exons here of RUNX1. And then the breakpoint can be close to an exon or further down in the intron, but it is in that large intron. Anecdotally, what we also noticed in the few cases we had was that the breakpoint in ETV6 always seemed to be in the same label or region so far. So you can see, so these are the same patients as the previous slide, but this time highlighting the breakpoint on chromosome 12, and you see the last label that maps to chromosome 12 is always this 5th one of this small pattern that is indicated by a red arrow. And so by OGM, and so the last label on 21 and then this first label on 12 is typically separated by this darker area where the breakpoint lies. So when we got yet another patient, so just to put in some context, so this is where we obviously the patients are, we haven't been running OGM clinically, so the patients are still getting chromosomes and fish as standard of care. And most of our pediatric leukemias are enrolled on the Children's Oncology Group or COG protocols. So we have mandatory fish to perform. So this patient had ETV6 RUNX1 fish. So those of you who are not familiar, we use a single fusion extra signal strategy, meaning that the probes are designed so that the derivative 21 of the translocation, which harbors the driver event, has a fusion signal, a mixture of ETV6 and RUN, so yellow signal, whereas the derivative 12 of the translocation has a dim 12 signal. So this is the pattern you see on the left. There is some debris here that you can just ignore, but this is the chromosome 12 with the small red RUNX1 signal. And you notice there's no green because the green is entirely translocated to the DIR21, and this is by design of the probe set. Labs who deal with ETV6 RUNX1 testing know that a well-known secondary abnormality is gain of either an extra copy of the normal 21 or an extra copy of the derivative 21. And we did see an extra gain of the normal 21 on this patient. We also did see a subset of cells, about 10%, where we had no signal on the DIR12. Now, given that this translocation is cryptic by chromosome analysis, by standard of care testing, we could not resolve whether or not it was just an artifact due to the dim signal being lost for some reason, or if there was a further rearrangement of the DIR12. Anyway, so when we ran OGM on this patient, we were expecting to see the 1221, which we did, so fantastic. But when we took a closer look, we realized that the software was not actually calling an ETV6 RUNX1 fusion. It was calling the 1221 translocation and did recognize that one of the genes did overlap with ETV, the breakpoint did overlap with EV6, but put the breakpoint outside of RUNX1. And you obviously here you can see that there's something else going on chromosome 9, some deletion that we actually did see by chromosome analysis, and there also seems to be a rearrangement between 9 and 12. This is not the focus of today's presentation, so pardon me if I'm going to skip over that part and just focus on the ETV6 RUNX1 fusion. So when we took a closer look at the OGM data, we noticed a few things. First, that on the DIR21, which is the driver event, so this is where you expect the ETV6 RUNX1 fusion to happen, we did notice that one label mapped to both chromosomes 12 and 21, and also that there seems to be some collapsed label on 21, which made us wonder if this was causing the software to miscall the breakpoint. So if we look at, and just for reference, this is the label where normally the breakpoint is in the cases we've had so far, meaning that this typically is the last label that maps to chromosome 12 and typically downward on the consensus molecule, you get into RUNX1 or chromosome 21 material. If we take a quick look at the ETV6 gene, so again, this is the label where that typically is the last one to map on chromosome 12, at least in the cases we've had so far. You see that, it seems things have been shifted by one label in this patient. However, if you look back, the breakpoint, if this is indeed on chromosome 12, the breakpoint would be somewhere, so that could still fit with the breakpoint being before the last 3 exons on ETV6, as is reported in the literature. So going back to the chromosome 21, so it's not really clear whether this label does map to 12 or 21. We suspect it maps to 21, and therefore that would put the breakpoint here, which again would be consistent with the literature. So this is one example where unfortunately the OGM data, it doesn't quite resolve where the breakpoint is. Now, being said, from a clinical standpoint, because OGM is not clinically validated yet in our lab. We are still using the FISH data for the standard of care testing. And for the children oncology group protocols, this is the standard of care. So demonstrating diffusion by interphase and metaphase FISH is sufficient to classify this patient as having an ETV6 RUNX1 fusion, so the patient was treated as such. And certainly the manual review of the OGM data supports the presence of an ETV6 RUNX1 fusion, even if the software actually puts the breakpoint outside of RUNX1. So I guess what I wanted to illustrate today and with this brief presentation was that, as any wonderful emerging technology, there can be a bit of hiccups. And so OGM does not always properly call the breakpoints of gene fusions or other types of rearrangements. So it is important to manually review your data to determine if the breakpoints make sense, both in terms of what is called by the software, but also in terms of does it make sense with what we know about the biology of the disease, because certainly we are aware that there can be mimics where you seemingly have a translocation involving 922, but that actually doesn't lead to a BCR-ABL1 fusion. And so it's something to keep in mind. Be aware that if the breakpoints are close to the 5' end or the 3' end of the gene, depending on the density and space between labels, you may get false negative results in some circumstances. So it is important, particularly if you're using if you're filtering only to show SVs or CNVs that overlap with certain genes of interest, you have to make sure you have the proper buffer in terms of overlap to capture cases like these where the breakpoint is wrongfully called outside of the gene. And certainly this work would not have been possible without our collaborators, both our postdoc fellow, Alexandre, as well as our collaborators at CHIO and TOH, and the GDL staff who performing the clinical testing. So that's all I had for today.
Great, thank you very much. Very interesting case. I see Adam, he has his hand up.
Hey, Mel.
Hey, Adam.
Could you show me the circus plot for the 9/12/21?
Yeah, I knew it's like, oh Lord, if I put that slide in, they're gonna ask about it. I decided for transparency and say this is what we have. Yes. And this is the challenge with BAL. I find that you get more than you bargain for. Yeah. I mean, it's by chromosome analysis, we did see what looked like a deletion, well, a terminal deletion 9p. And there were also other losses, deletions that we saw by chromosomes and duplications that we saw by chromosome and OGM. But yeah, it's, there's also this gain on 12p. So I suspect that what looks like a del 9p by chromosome is probably some sort of unbalanced rearrangement between 9 and 12, where some part of 12 has been duplicated, for lack of a better word, and I don't know, either copied there or inserted. I don't know.
Yeah, I mean, this is one of those things, I think, where you have to pick one end of the rearrangement, and then you have to take each hybrid molecule and follow it into the next one, because this was a little bit of what I was concerned about. I'm not sure what the breakpoint is on 9, but it could be an ETV6 rearrangement with another partner on 9, like PAX5. So it could actually be PAX5 ETV6, and ETV6
well, so
is
no, so the breakpoint. So when we look at the data, this rearrangement with chromosome 9 does not involve ETV6 at all. It's way down, so you can't really appreciate it on the circus plot here. But it is way down, so it does not involve. And also the VAF of this 9/12 is much lower than for the 12/21. And if you look at the 9, you also see that the copy number is not the same. So I suspect that we have some clones with different sizes of deletion 9, some of which may have been further involved in the rearrangement with the 12. But it does appear this 9/12 rearrangement does appear to be subclonal, because the VAF for the 12/21 is 0.45, 0.48, and the VAF for the 9/12 is 0.25, if I recall correctly. So much lower. And the deletion 9 by chromosome analysis, if I recall correctly, was also subclonal, not seen in all the cells. So that's the challenge.
It is very challenging because
you see more than you want
we've
to.
seen some, sometimes we had a case of an FGFR1 rearrangement that was rearranged with TPR1 on chromosome 1, but you couldn't see it directly from the OGM data because there was a rearrangement on chromosome 1 which flipped TPR1 to the breakpoint on chromosome 1/8 breakpoint. And so sometimes when you get into these more complex rearrangements, I don't like that particular phrase, complex rearrangement, but when you get into these multi-step rearrangements, that sometimes it really does, you really have to look at it hard in order to figure out exactly where everything went at the end of the day. But it's very interesting. And ETV6 is a challenging gene to look for rearrangements because not all the rearrangements with ETV6 are in frame. Some of them are just juxtapositional. So
yes. And also, and we know that you have subclonal deletions of ETV6 and ETV6, right? So sometimes there's lots going on, and again, remembering that OGM sort of, yes, in terms of detecting hybrid molecules, but in terms of copy number and things, you're kind of looking at averaging out things. So it is challenging. And from a clinical perspective, it's always, where do you draw the line at what do I report? How do I describe things? How much confirmation I do? So it is certainly one of those cases where you see more than you would like to see. So it's where and the challenge is that if you want to try to confirm these, we don't always have lots of material left on these patients to do all these fish. I mean, you can do microarray to look at the losses and gains and to see if they confirm what you see by OGM, but for these rearrangements, you're kind of stuck with trying to do fish on whatever fixed pellet or slides you have left on these patients. So sometimes it's really challenging, especially when they have low VAF and you have no clue if you're going to be able to see them. So it's OGM is both beautiful and overwhelming.
Yes. It's fun times ahead, everybody.
Yes, and I our experience so far has been that BALLs are more complex than AMLs, and so I applaud anyone who went live clinically. And because certainly with most of our cases, pediatric or BALs, and we have seen some that are quite complex to the point where you're kind of pulling your hair and saying, oh Lord,
that's been our experience as
how
well.
do I start? Yes, exactly. And there's obviously you want to focus on what's clinically relevant, like ETV6 RUNX1, for example. But at times physicians are interested in knowing if there are subclonal abnormalities that could predict relapse. And so again, it's a matter of where do you draw the line, and it's only focusing on abnormalities with non-clinical significance is a moving target because we keep discovering new ones.
Great, thank you very much for that interesting case presentation. Just cognizant of time, and actually we have time to get Victoria some opportunities to talk about her experience with EQA. So Victoria Marcu, are you available? Check through the list here.
Yes.
Great.
Just share my screen.
Great, thank you very much for coming today.
So I was so now you can see my screen, right? So I have seen that you plan to speak about external quality control in this meeting, and I offered Adam that I will share our Israeli experience of just finished external quality control round trial for 2023. So the arrangement of the trial was of 5 participating medical centers. At every center actually provided shared RVA file, downloaded file of one case. So we took about 2 weeks to contribute the files, and I will just go back to what the files everybody distributed. And we agreed to have 2 AML cases, 1 pediatric and 1 from the adult patient, pediatric TLL, adult BLL, and the MDS AML case. And this was our chart for submission of results. So every case had a separate Excel sheet for every center to provide the detailed results. And you have every column should be filled, but you can everyone could add the rows. And the instructions were quite general. So we suggested to analyze with filters from the framework paper, but we actually wanted to see how everyone analyzed normally using nationally agreed bed files for ML and ALL. Those ones we just shared with each other and combined around a year ago. We urged everyone to use regular workflow for analysis to actually understand whether our workflow is giving the same results as we would expect from clinical testing. We wanted to, or we agreed to report tier 1 and tier 2, or any other findings that lab would usually report for hematology case. We agreed for 6 weeks for submission of results. So we had every lab sent their results to one laboratory from Rambam Medical Center that volunteered this year to take care of organizing the data. And those were our cases. And actually, I chosen to analyze the data or success of the trial in a number of structural variants or copy number variants that we agreed that should be reported for every case. Like for this case was the 4 variants, obviously this translocation, trisomy 6, trisomy 19, and trisomy 21. 2 laboratories reported additional findings, and that afterwards were agreed to be actually not necessary. And that were our cases. So as you can see, the cases are not very simple. And actually the case of MDS had this chromoanagenesis or chromoplexy event between chromosomes 2 and 3 that several laboratories reported in a different way, and some even reported it as a long list of separate event describing every fusion and loss from on every chromosome. And so there was actually difference between the groups. And for 2 other cases also, we had actually 2 slips from different labs that did not report, one of 3 events here and one of 5 events in this case. But most of the reports I calculated concordance by just counting all number of events that everybody should be reporting, and I got so we had 83 reports out of 85, and this would be 98% of concordance for the reporting. And so this, the conclusions from this were that so the summary of results took around 2 weeks, and then we had the Zoom meeting. Israel is a very small country, but we are very distant centers, and we meet every month via Zoom. And we discussed in the last meeting the results, and we saw that minor changes in filters definition may very much influence the result, and in reporting, non-reporting decision, and chromotripsis, chromoanagenesis, and chromoplexy events, which I'm not sure what is a very exact difference between all of them, and when and where I should use each of the terms. But we decided that all of these events should be reported as one finding, and not a long list of fusions. And that's just to make simple statement for the clinicians that receive our reports. We agreed to combine sequential gains and losses in one-to-one reported CNV. Like here, you can see that the program would report two separate gains, but or 3 separate losses, but those should be reported as one loss and one long gain. And and in the interpretation, we can indicate for this case, it's probably either chromosome 5 for Q arm. And if we have a loss, program suggests also the fusion for every loss, and this should be reported as a loss event. And not everybody were familiar with ISCN and gene fusion nomenclature that was just ratified. And we had to make a decision just in some decisions for quality of our control trial, that it was not very clear which frequency, what is the frequency threshold for the reporting. So event of frequency of VAF 0.09 should be reported or not if it is not significantly fusing known genes, and there was also discrepancy between the groups. We also have still not clear consensus about copy number, variant size, which are not covering bed file genes for reporting event of 1 megabase pairs of 5 megabases should be the threshold for reporting. We see in many cases copy number variants in IGH locus, and we agreed not to report them, but I'm not confident whether it's the right thing to do. We agreed to revise and update bed files, including coordinates that was mentioned today, that coordinates not always are very accurate, and directions of the genes exactly for the decisions of significance of certain translocations and involvement of genes in those translocations that we see with OGM. And so there are a lot of questions were raised, but in overall, every participant, and each group has a different amount of personnel that is was involved in this analysis, but everybody were very satisfied with this program and found it very productive and contributing. And of course, according to everyone's accreditations, every group was responsible to summarize and decide about the results of their own participation in this trial. Because we cannot give grades to each other, and actually we are not sure yet which are true or fail answers for. And these are two important papers which we agreed to follow for ISCN nomenclature for OGM, and adapt as much as possible framework paper guidelines for the analysis. So that was our experience, and you can, if anyone has any questions, you're.
That was fantastic. Thank you very much for sharing your experience. It's very topical for some of us getting close to or currently reporting tests, because this is where we're now looking, is what do we do? And I think you guys have developed a nice framework for how that works, and it sounds like a really useful model. I also want to say I love your Israeli OGM forum logo and just picturing it with a maple leaf in the middle for Canada. That's great. So I'm cognizant of the time. So I just wanted to say a couple things first, and then I'll let people know if they're going to ask questions. I understand there's been some inquiry about whether the pre-survey, pre-meeting survey is still available, and it is. Jenny has informed me she'll send that out post-meeting, and feel free to contribute to that if you haven't already. And I do also want to send a link in the chat here to another short survey, just asking if you're interested in participating in EQA in Canada in a similar kind of framework, just to get a sense for who wants to have this discussion and what kind of samples you're looking for. So please feel free to fill that out when you have time. I'll send it out afterwards. But are there any questions for Vika? Vika? Okay, so I actually was going to ask you if you wouldn't mind sharing the Excel sheet that you use to tabulate the results. That would be helpful for designing a similar kind of process. I have a rough draft of something that's not quite as nice as what you've shown us. So something that's already been worked through and works well would be very helpful.
Yes, I can send it.
And do you find, is everyone using the same kind of access build and informatics pipeline right now, or is there variability?
Excuse me?
So I know some of us have, like we're currently on Access 1.7 and have a different solve pipeline than others in Canada. Is it similar in Israel? Is everyone on the same kind of access version and solve version?
Yes, there was actually this the program was done in between the upgrades. So some labs underwent the upgrade and some others did not. And actually contributing to the discussion that was here today, I also have mentioned not very detailed, but I see that samples that were run in version 1.7 analyzed a little bit differently in 1.8, and actually one of the slips that were so one of the labs did not see one of the structural events that everybody else were reporting because of these differences in the version of the program that they used. And after the upgrade, they opened the same case. Have seen that very clearly. So it is possible that some changes between the versions are showing us more information that we know and we have seen before the upgrade.
Interesting. Was it a critical structural variant, like a
Like
driver?
Icarus deletion or something like that.
Oh, interesting.
Yes.
Interesting. Yeah, we'll have to watch for that when we go ahead, which we already see a lot of those. I'm surprised there's any missing, but that's good to know.
The Icarus deletion, I think, is a bit tricky depending on where you set the population filter threshold. The group from Belgium that was doing a lot of BALL, this is Barbara Diwali and Katrina Rack, had said that on some of their analyses using older versions of the software, they had to increase the population threshold to 2% in order to see some of the Icarus deletions. That may be, there may be something in the population database there that was overlapping, that was maybe hiding some of that data. So, or it may just be a consequence of the cancer cell fraction in those samples.
That's possible, but the new version actually solved this, I think, at least for that case specifically.
Interesting. Melanie?
I had a quick question. You mentioned being some disagreement between labs regarding the size of CNVs to report when it's not overlapping with a bed file. So you mentioned 5 megabase, which I think is what is in the framework, but you also mentioned 1 megabase. So I was wondering, what about, something in the middle, like 3 megabase? Is there any agreement, or you're all over the place?
Well, so I think that most of us agreed that the framework decision about 5 megabase should be the right one, but actually from the other side, if we say that karyotype resolution is 5 to 10 megabases, then what is the advantage of OGM if we can see big copy number variants and 1 megabase or 3 megabase is a huge variant. So I don't know what is the correct answer, and is there a correct answer, but this is something that the only thing that I think that the 5 megabase copy number variant is good, that we can normally prove the presence of this variant by other methods, because we sometimes not sure what is it real or what to report. And of course the variant allelic frequency is also the player for the decision of those. And so it's under discussions, and we are a small group, and we'll be glad to hear opinion from the bigger audience, of course.
I think I should probably comment from the perspective of the framework. And the 5 megabase cutoff in the framework paper was chosen primarily to be in alignment with the ACMG recommendations for neoplastic testing in microarray settings. So microarray, both the ACMG guidelines and European guidelines set this 5 megabase cutoff. Now the 5 megabase cutoff is when you're evaluating a genome for the presence of complexity. So you're looking for cytologic or cytogenetically visible alterations that are larger than 5 megabase. They can be counted towards genome complexity. Okay, so that was sort of where the 5 megabase comes in. Now, we didn't say that you couldn't interpret copy number variations below 5 megabases. the big problem that we have with doing this kind of interpretation is that we don't have a lot of data to go back to inform about the clinical significance of those. And so this is where I think we recommend the use of region files that overlaps genes of significant interest in the disease in question, in this case, usually hematologic malignancies. So defining a region file that calls out or pulls up these results so that you can then interpret them for their pathologic significance when they're below 5 megabases. So like if you have a very small TP53 deletions below the 5 megabases, we're not saying don't call it. We're just saying, you know, if you have a 17p deletion that doesn't involve TP53 and one that does, that you can use those region files to assign a tier to that particular abnormality. So yeah, there's nothing in the framework that says you can't interpret anything below. Our you know, our strategy was also though to help to minimize reporting of tier 3 events when necessary, because what are you going to say about them? There's going to be, you know, a significant number of them in every case. So our idea was to try to minimize the reporting of them as much as possible until there is information to support the elevating, for example, the clinical tier for recurrent abnormalities.
Yeah, I agree. I know that there is always the compromise between what we see and what to report, and what the clinician will do with our report and with the information we provide. It's all still should be decided. But I think that now we have a lot of information that we one day retrospectively we can analyze clinical significance of all of it, and probably be smarter in a few years.
Great, thanks very much. That was a great overview of what you guys are doing, and just cognizant of time here, so I just want to remind everyone, hey, thank you for coming. It was a great discussion today. A lot of really informative things we can think about. Please complete the CPD evaluation and feedback form if you want a CPD certificate and to help improve on further sessions. Like I mentioned, the survey that we send out pre-meeting will go out again post-meeting, and I'll also send another survey that is geared more towards Canadian EQA initiatives. I know I've discussed this with some of you, and I'm sure others have discussed it offline as well between themselves. So I think it'd be great if we can come to some kind of consensus about how to do something similar to what's been done in Israel. And I think that's a really good framework to work from, but how we can best design it to fit for Canadian labs and what we need. So we kind of ran out of time today, but I'm happy to take that discussion offline or schedule another meeting to talk about that more specifically if you guys respond to that request for, you know, solicitations of interest. I understand there's also plans to talk about this at the OGM user group meeting coming up in a little over a month. So I hope to see some of you there and chat more about it then. Thanks to everyone who presented today. And we'll plan to do this again before summer, hopefully, and reach out in advance to ask for more cases, topics for discussion, anything else. Anything else before we close up for the day? Great. Well, thanks everyone for your engagement, and talk to you soon.